Hello students, in today's class we are going to discuss about few more enzymes that are used in cloning reactions or in recombinant DNA technology such as polymerase, kinase, phosphatase, nuclease, terminal deoxynucleotidyl transferase and reverse transcriptase. So the first slide talks about the activity of enzyme alkaline phosphatase. So here we have our vector molecule that is digested with the restriction enzyme, and we can make use of alkaline phosphatase to remove the phosphate molecules that are present on the vector. Now when we try to ligate this, then it cannot self ligate uh, because the this can self ligate, but after removal of the phosphate group it cannot self ligate on addition of ligase because the phosphodiester bond cannot be formed. Then we have our insert molecule, that is a normal insert molecule containing phosphate at the 5 prime, hydroxyl group at the 3 prime, and we mix the insert and the vector in presence of enzyme DNA ligase and ligase buffer. So we will have formation of one phosphodiester bond on each strand, and one nick will stay on each strand here since the phosphate is missing. However, we do the transformation reaction as such, and after transformation the host repair system will automatically repair the nicks that are present on each strand. So in this way we can make use of enzyme alkaline phosphatase that will ensure that the recircularization will not take place without the presence of the insert molecule. So that improves the chance of isolating the recombinant DNA. Then the second slide talks about homopolymer tailing. So for homopolymer tailing, first we need to treat our insert as well as vector molecule with an enzyme lambda exonuclease. So this exonuclease specifically chops off bases from the 5 prime end of DNA molecule, and this results in generation of a 3 prime overhang, which is a requirement for the next enzyme that we use in homopolymer tailing, that is terminal transferase. Terminal transferase enzyme can add nucleotides, template independent nucleotides towards the 3 prime end of the template DNA, and it will add whatever nucleotide is supplied to us in the reaction. So if we provide ATPs, then a tail of adenines will be added at the 3 prime end of the vector molecule, for example. Similarly, the insert molecule is also treated in the same fashion, except instead of ATP molecules, TTP molecules are used as a substrate so that a tail of thymines can be generated. So we basically have complementary tails being added to the insert and the vector molecule respectively. The two are then mixed and annealed, followed by a ligation reaction, which will, so since we are using different types of tails here, it will allow complementary base pairing and more chances of getting recombinants. So there are some points that are written over here about the activity of enzyme terminal deoxynucleotidyl transferase. Then coming to the next enzyme, that is polynucleotide kinase. So polynucleotide kinase, this enzyme, what it does is it can add a phosphate group from the substrate, that is ATP, to the 5 prime end of a dephosphorylated DNA molecule or RNA molecule. So what we do is first we need to do a dephosphorylation reaction, and then so that a hydroxyl group is present instead of a phosphate at the 5 prime end. Then we add the enzyme, its buffer, and in presence of ATP molecule, we will have addition of a 5 prime phosphate molecule. Generally, we make use of radio label phosphate here so that your DNA that is generated or RNA that is generated is radio labeled in nature. So this method is basically used for generating labeled DNA or single stranded DNA or RNA probes that can be used for southern blotting or northern blotting technique for detection of specific DNA or RNA molecule. Now coming to the next method, that is cDNA synthesis. cDNA basically stands for complementary DNA. It is basically a synthetic DNA that has been transcribed using messenger RNA molecule as a template using the enzyme reverse transcriptase. So DNA contains both coding as well as non-coding sequences. However, cDNA contains only the coding sequences. So for cDNA synthesis, three different strategies for priming are being used. The first method is a random priming method. So in a random priming method, it is basically used when we do not have any information about the template. And we basically make use of shorter primers, which are about 10 base pairs in length. So the chances that these find sequence complementarity and ability to bind will be higher. However, the binding will be at random locations. And it is also difficult to have full length amplification of the cDNA by random priming method. The second method here is by making use of oligo DT primer. And this method is based on the fact that the eukaryotic messenger RNAs have a poly A tail at the 3' end, and exploiting this, poly T primers can be used. So here again, what will happen? The total messenger RNA of the cell will be converted into complementary DNA. While the third method is applicable, sequence specific primers, this is applicable when we have some sequence information about the gene that we want to convert into cDNA. So the primer can be synthesized since we already have the sequence information. So here you can, since the primer is specific, so you can study specific genes. Their presence, absence, expression can be understood by cDNA synthesis using gene-specific or sequence-specific primers. Now coming to the methodology for cDNA cloning. So we need to also process the plasmid, but first we'll discuss about processing of the insert molecule. So here we have the messenger RNA molecule that acts as a template, and we are using poly DT primers over here. So they will bind at the poly A tail. Then we add the enzyme reverse transcriptase. So reverse transcriptase is the enzyme present in retroviruses. So it will synthesize DNA by making use of RNA as a template, and we have formation of the first strand of DNA. After this, we do alkali treatment, and also because of the RNase H activity of reverse transcriptase, later on the RNA strand is degraded, and we are left with a single stranded complementary DNA. Now we only have one strand of DNA here, and the other strand also needs to be synthesized. For synthesis of the other strand as well, we need a primer, and the primer for the second strand synthesis is basically dependent on a self-priming reaction which is formed by formation of the secondary structures in the cDNA that generates a 3' end which will mimic the 3' end of a primer, and the template is also there. So in presence of enzyme DNA polymerase and the building blocks, the second strand synthesis will take place. Now we are having double stranded DNA, complementary DNA. However, at one end we have few bases which are not base paired or which are single stranded. So we make use of enzyme S1 nuclease, which specifically acts on and degrades single-stranded DNA molecule. This results in generation of the 3' and 5' ends on both ends of the insert molecule or the complementary DNA molecule. We can then make use of enzyme terminal transferase, which will add complementary cytosine residues on the insert molecule. And similarly, we digest our plasmid molecule, and after treatment with terminal transferase, we add the complementary guanine nucleotides at the 3' ends of the vector molecule. After mixing or annealing the two, we carry out the ligation reaction in presence of enzyme DNA ligase, followed by transformation and screening of the recombinants. So in this way, cDNA cloning can be performed by first converting the messenger RNA into complementary DNA, and further cloning is done by the standard procedures. The points for cDNA cloning are mentioned here for your reference. Next enzyme is DNA polymerase. I have another diagram for DNA polymerase which I would like to use. So DNA polymerase enzyme is basically involved in a PCR reaction that is used for amplification of your template molecules. So there are different stages to PCR. So the first step is initial denaturation reaction, which is slightly elongated than the cyclic denaturation reaction. So after initial denaturation, we have separation of the two strands of the template DNA molecule. Then the temperature is decreased at around 55 degrees Celsius, where we have primer annealing, where the forward and the reverse primers will bind to the two template strands respectively. Then the polymerase will attach to the free 3' hydroxyl end of the template molecule and carry out extension at about 72 degrees Celsius, completing the formation of the total DNA strand or extension of the fragments. This is then followed again by the denaturation step, and the steps are repeated about 30 times to complete the polymerase chain reaction or extension reaction. This enzyme is basically isolated from a bacterium which is known as Thermus aquaticus, which is basically a thermophile, because of which the enzyme is active and is able to be active at very high temperature of about 94 degrees, which is used during the denaturation step. So there are several applications to this enzyme. First of all, it is used in a PCR reaction. It is also used in some methods of DNA sequencing and can be also applied for making DNA probes. So that's it about different enzymes that are used in cloning reactions. Thank you very much, and kindly like, share and subscribe if you like the content.